morning again. Continuing this, this is, so the previous one was part of the C5 course, I think it was. And this part belongs to another course, which was C7, that has, it was something about mechanical and electrical properties beyond AFM. Okay, so just trying to see outside the use of, oops, okay, about the use of other techniques that also can be used to investigate the biological systems, but which are not strictly related to atomic force microscopy. Okay, so the idea of this is to give a complementary background so that you have not only information of one specific technique, but of some other technique. So the reason I'm giving this is because I also work with other techniques which are not AFM, so then I can present some results, which is just, but to start with, I will go back in time, trying to figure out what has been the historical development of electrical techniques in biology and in medicine. And for that, we had to move to the field of bioelectricity. And these dates back from the days of, oops, it doesn't work now. I think it doesn't have, okay, no, it's okay. Okay, so the idea is in these old times, it was demonstrated that if you take a dead frog and you connect the dead frog with two metal wires that have different composition, then it was found that when you put the connection in two nerves of the frog was responding. And this was historically like the first demonstration that electricity and biology are intimately related. And therefore, this was the starting of the field of bioelectricity in the sense of electrical techniques relevant for biological functioning of living systems. You can go also back to the old times where the first medical applications of bioelectricity were developed. And among these medical techniques, of course, there is the electrocardiogram, which is basically recording the electrical activity of the heart by using some external electrodes. You see, these setups has nothing to do with AFM, so it's completely different. But they provide information that is also very relevant for applications. Okay. So what are the main players in all this bioelectricity? So in this second part, I will be spending more times on active properties. I will present something about passive properties, but also I will consider also active properties of electrical systems. And basically, in bioelectricity, the main players are the ions, and these ions move from inside to outside of the cell through ion channels, through other specific proteins, which are called transporters, which are able to change also ions from the inside and outside of the cell, and the membranes that separate the different cell compartments, and in particular, the cell from the outside of the cell. And all the electricity that we have in our body is generated by this mechanism. Basically, you have ions, ion channel, transporters, and membranes, and all the metabolite processes. But this is how the electricity is generated. And therefore, over the years, there have been different techniques trying to measure these electrical techniques. So if we put in terms of physical quantities, because we would like to measure quantities here, so there is a number of quantities that are relevant in bioelectricity. As I mentioned before, concentration of ions outside of the cell, for instance, and inside of the cell, they are different. And this is the key point. So keeping a different concentration, this is the battery of our bodies, because then when there is an ion channel that opens, the concentration tends to equilibrate, and this produces fluxes of ions, and this gives rise to the electrical activity of the cell. And other quantities that are relevant is the conductivity of the media. So how many ions do you have inside? Electrical conductivity, how many ions do you have outside? And also the polarizability of the different media. Now I don't need to explain what is the polarizability because I have explained it in the previous presentation. But basically here you have the polarization of the inside of the cell, the outside of the cell, and then in this case, just to make very simple, the polarization of the cell membrane. And then you have current fluxes, ion fluxes going in and out, and you have electrical potentials in and out and at the membrane. Okay, so these are the physical quantities that are usually present in bioelectricity phenomena, and these are the ones that the different techniques try to address by using different techniques. So now here, let's try to distinguish what is passive and what is active. As I mentioned before, in the previous presentation, I was talking all the time about passive electrical properties. What I understand by passive, well, basically, if you have here is the passive, if you have a cell, and then if you put into an external electric field, so an electric field generated by some electrodes, then the passive response tells you how these electric fields interact with the cell and produces a response in terms of electrical response of the system. So if you, for instance, if you apply a voltage, then this will produce a current, and this current will depend on all these properties. But the values of these properties do not change due to the external field. Okay, so this is the basic assumption of the passive properties. Okay, so the material does not change the properties when you apply an external potential electric field. Instead, in the case of active response, you don't apply anything to the cell. So the cell is there alone. And then there is something that activates the electrical response of the cell. This can be a chemical substance, for instance, a neurotransmitter arrives to the cell, opens the cell, the ion channels, the membrane of the potential changes, then the voltage gated ion channel activates, and then you have a response of the cell. So in this case, in the active response, the cell is the source of electricity. Itself, it generates electricity, and then externally, we put just some electrodes, some system to detect the electrical activity generated by the cell. In this case, the properties of the cell changes, some of them changes during this process. For instance, basically the conductance of the cell membrane changes during an action potential. So there is an active change, and this is what gives to this active response. Okay, so then depending on what is the situation, we will measure active responses or passive responses of the cell. So what is the setups to measure this property? Basically, we need electrodes. That's the idea. We need some sort of electrodes to put there. Of course, these are different historical, so this is not how we work today, but basically you need to generate, for instance, a potential to apply to a cell and then to measure a response. So you need to generate the potentials externally. In the old times, we used the Leyder jar. So we are talking about 200 years ago. And these were able to generate potential differences. The big advance was the voltage battery. This was the first battery that could provide a constant potential applied, and this was what helped a lot. And then there is many other systems. And of course, nowadays, we use source meter apparatus, which are instruments that are able to apply voltages and to measure currents. And the electrode is normally, or some of the electrodes we use, typically they are glass pipettes. And inside the glass, you put a silver nanowire, a silver chloride wire. And this is the one that you use to detect the currents. Okay, so these micropipettes, they have an electrode inside, and this is what you plug into the system in some cases. In some other cases, you put other type of electrodes, but this is just one example. So then apart from sources of electricity, you need meters. So how to measure the electricity that is generated. Of course, we have old examples here, and then the arrival of electronics give us the amplifier, and this is the main core that allows us to amplify electrical signals, and all these amplifiers now are set up into the same mother source units, and these ones are the ones that amplify the electrical signals and allow you to measure them. And finally, also very important is, beside these two techniques, also microscopy is also very helpful, because with microscopy, you can look at the a structure. For instance, this is a neuron design where you see what is the system that you have, and then where you can put the electrodes to measure different properties. So what is the setup basically nowadays that is used to measure beyond AFM to measure electrical properties in cells? Well, the gold standard, if you want to measure active properties, is a technique which is called patch clamp. Okay, patch clamp is basically a technique that uses a glass micropipette, which has a diameter here of a few micrometers. This glass pipette is then plugged in contact with the cell, and inside the pipette there is a silver chloride nanowire. Wire is not nano, and then basically you can measure the currents or the voltages that are generated by the cell. Okay, and of course you need to set up in order to make this contact. Of course, you need a microscope where you, by micropositioners, you go there and you contact the cell, and then you can measure both passive and active electrical properties. So this is one type of setups. This is mostly for active properties. And then you have planar microelectrodes, which is the other type of system that is used to measure electrically. In this case, you have a planar electrode, and then you put the cell on top of it. And then you measure the electrical activity of the system. But contrary to this case, in the case of planar electrode, in this case, you puncture the cell and you open the cell membrane. So then you can have the access to the inside of the cell. So you can perform what is called intracellular measurement. With this one, you only measure from outside of the cell, and then you measure what is called extracellular potentials, which are different from the intracellular. There is a complicated relationship between one and the other, but still you can monitor the electrical activity of the cells by using these techniques. Okay, so what are the type of signals that you get? For instance, if you have a cell, a neuron, and you inject a current into the neuron, if you don't reach the threshold for the neuron excitation, basically you have what is called the passive response of the cell. This is the voltage of the membrane. So the voltage of the membrane increases to a certain value, then it decreases. So this is a way to measure variations in the membrane potential. So you excite with the current, and you can tune the membrane potential to the value that you would like to have. If the current injection that you put is high enough, then you will activate voltage-gated ion channels, and then you will have the action potential. And this is the signal that travels across the nerves in our body, and that carries information from the brain to the rest of the body. Okay? So this signal, again, can be measured or by patch clamp, but mostly many experiments are done on specific types of neurons, which are very, very big, which is called the giant neuron of the squid, for instance. And in that case, you can put an electrode inside the neuron. It's that big that you can put an electrode inside and another outside to measure the intracellular potential. In human neurons, this is not possible. So you don't have a space to put the intracellular potential, and therefore, this is normally measured by using patch clamp techniques or other types of techniques. Okay. And this is the intracellular part. If you put an electrode, a planar electrode, the signal that you get is something like this, which is not exactly this one. It's very different. And this is what's called field potentials, which are the potential that the cell generates in the surrounding. So one thing is the difference in potential between inside and outside, and the other thing is how this potential propagates then outside of the cell. So this extracellular potential typically can roughly demonstrate that they are related to the time derivative of this other signal. So it's more complicated than this, but this is just to tell you that extracellular potential and intracellular potential, they have different information. And therefore, you need to know these type of things. More things that you can do. For instance, here, in this case, what you do is you apply a voltage to the membrane, and then you measure the current evolution. This is currents. I don't go into the details, but this is the current generated by the cell itself. So this is active response. Or You can do, for instance, impedance spectroscopy, which is a passive response. So basically, you put the cells on top of the electrodes, you apply an alternating voltage, and by analyzing the response, you have what is called the complex permittivity, which is related to the permittivity I introduced before, but there is a lot of stuff there to be understood in this experiment. Or even if you want to go to molecular level, even it is possible to record the activity of a single ion channel by using, for instance, the patch clamp technique. And in that case, the major finding is to know that the ion channels are behaving randomly. So the ion channels that generate the electrical signals, they are random generators of electricity. And our signals at the level body is the average of all these random behaviors. So this can be measured by using the patch clamp technique. Okay, so this is again an active part. Okay, so then this is the standard, what is there. So then I wanted just to show some new techniques that could be of interest to maybe some of you that go beyond this is what is the standard. Okay, just to give a flavor of what are the directions that the people is, of course, is not comprehensive. There is much more than that. This is just to show some few examples. So the first example is what we call, well, what is called impedance flow cytometry on a chip. Okay, so this is what is used for. So basically, in this case, what we would like to do is to count the numbers of cells that passes through a microchannel. I think I saw the other day in some of the presentations, something like for mechanical testing of the cells or something like this. So it's exactly the same concept, but rather than testing the mechanical, you would like to test the electrical properties of this cell moving through the channel. Okay. And basically what you do is you have a flow so that this can be a cell, or it can be a liposome, or it can be other type of objects. And what you would like to see is first, you would like to count how many of these are passing by there. And the other thing you would like to detect is a bit what is the passive properties of this object, because you would like maybe to distinguish different types of populations inside this flow of particles there. Okay, so this is there is an apparatus which is called Coulter counter, which is used in most of the biolabs, which is used to count cells. This is, let's say, an on-chip version of this apparatus plus some additional features. Okay, so basically what it can be done, well, this is some of our work, but many others I insist have done similar things. So basically what we do is you inject here the cells or the particles that you want to detect, and they go into this microfluidic channel. And in order to count them, you would like to align them one after the other. So then we use a property of the fluids in microchannel, which is called laminar flow. In the laminar flow means that you put two fluids in a channel, they don't mix with each other because there is no convection, because it's not turbulent. And therefore, basically what it does if you put two lateral flows is squeezing the central flow, and they don't mix at all. And basically this is what you see in this drawing. No? Basically these two lateral flows are used to squeeze the central flow so that the nanoparticle, so the cells will pass one by one. And then you put two electrodes. These two electrodes are connected to a voltage, an alternating voltage, and therefore you every time it passes between two electrodes, you detect a change in the electrical current between the electrodes, and this is related to the properties of the cell. So this was a video, but I think it is not synchronized, unfortunately, but just for you to show. So basically, well, this is the video. You have seen something passing by. I can repeat because it had to be a 10. Okay. So there will be two particles passing by, one now, and now it should come another one at some point. This is a high-speed camera, so this is a slowdown. And basically, every time a nanoparticle passes here, you get this type of signal. And then you have this type of signal. Okay. So then this allows you to count how many cells are passing by. In our setup, we are not the best by far. we could count even 1,000 cells per second because it's very fast and the electronics is not specifically fast. And what is relevant for us was that the size of this peak is related to the size of the particle. So we can count, we can size. And then some other groups have added some additional functionalities. Look, now it's working, which is, for instance, trying to separate the different types of red blood cells and white blood cells, for instance, if count them in a flow. And basically here you have a distribution of sizes of the response of the system. And here a distribution for the white blood cells that, of course, they are much bigger and they give much bigger difference. So then you can differentiate between a red blood cell and a white blood cell, which counting one after the other and determining the size. So this is one technique. Here, the relevance of this technique is that it's on a chip. Okay, so the idea is that to integrate this function in a chip so that it's something that is portable, it's easy to use, it's cheap, etc. So then, if you look at the response of the cell due to this external electric field, this is passive response. Basically, as I mentioned before, the cell has intracellular properties, membrane properties, and extracellular properties. If you change the frequency of this voltage and you measure the current there, what you see is that the impedance, well, the impedance is the ratio between current and voltage, basically. The impedance depends on the frequency that you apply. And you see that you have different transitions, and every one of these transitions gives you information on one of the physical parameters. So it means that if you do a full spectra, you can get information on a population of cells. It's not a single cell measurement, but it's a population of cells. Okay? So then this is what it has been done by other groups. And basically, in this case, what they did is they use two frequencies. You use a low frequency here, which is basically giving you the size. And then if you use a higher frequency, then you can detect, for instance, the membrane capacitance of the system or the intracellular conductivity. And in this way, you put two frequencies, you can determine the size and give an additional property of the cell. And in this way, you can determine here, for instance, you measure the conductivity of the cytoplasm, the membrane capacitance, the electric constant of the membrane, and this is this is another factor, which is Clausius-Mosottis, which is different from this. But basically, what I would like to highlight is that here you see two populations that have different intracellular conductivity, meaning that the content, the ion contents of the two groups of cells is not the same. Okay, so here we are not doing at the level of a single cell with nanoscale resolution. It's a macroscopic measurement. But the advantage of this is that you can do 100 cells per second, which is in AFM is very far from what we get in our system. So a combination probably of the two types of techniques is what it will be interesting. Okay, so this was for passive properties. So now I will highlight some developments into active properties. So measuring the electrical activity generated by the cell. I will just go over different contributions from different groups and some of things we have done also. So one of the issues with patch clamp is that everybody likes it. It's the more reliable techniques in terms of the result that you get, but it's very invasive because you have to puncture the cell and you have to break the cell membrane. And this means that you have a limited time to do this measurement. You have a few hours maximum, and then the cell dies, basically. And sometimes, if you are not lucky, the experiment doesn't work, the glass pipette breaks down, then you have to start again. So it's a bit of painful technique. There are some automatic systems, but also they have their limitations. So people now is trying to develop alternatives, basically to patch clamp, that perform better than patch clamp. Okay, so then in here, for instance, what they have fabricated is a transistor. They put a transistor here in terms of integrated transistor. And basically the transistor was then immersed into the cell. You see, this is the transistor. It was put into the cell, and then they recorded the signal from the transistor. not from the pipette coupled to an external system. And what they have shown is that basically they got the same result as patch clamp, which was simultaneously measured. Okay, so this is just a technique that in principle performs the same as patch clamp. It has some advantages and many disadvantages, no? Because in particular, you have to be able to fabricate these transistors there, which is not something that everybody can do. Okay, but it's something that is alternative to the classical patch clamp technique. Another technique, it was, okay, rather than using a pipette, a micro pipette, which is also always very painful to use, we would like to use planar electrodes, okay? Because planar electrodes are easy to fabricate, cells can sit on them, and it looks something easy for us to handle. But then you get the problem that you don't have access to the inside of the cell. So people has developed a mixture of a planar electrode with some sharp needle that is getting into the cell. And the idea is that in this case, you have a silicon oxide pillar, which enters into the cell. And then here there is a transistor. This is a transistor again. And basically you have the source, the drain. In a transistor, there is source, drain, and gate. So the gate is the cell. Okay, so the potential of the inside of the cell is what acts like gate in this transistor. So when the membrane potential changes, the current that flows in here is affected by what is happening inside the cell. Okay, so this is a way to measure electrical properties of cells. And here you see the result when it is not inside the cell. So when the electrode doesn't puncture and penetrates, you see that you record a very, very tiny signal. This is called the extracellular potential, which are always very small. But when you enter the cell, you see that you start recording the shapes of the action potentials of this cell. Okay, so this is another way to measure active properties of cells by using novel technologies beyond just patch clamp technique. This is something relatively similar. In this case, we have nanowire electrodes. So basically, this is a nanowire. It's a sort of a metallic electrode. In this case, we don't have a transistor. It's just a needle. And the idea is that you can grow the cell on top of these transistors, and then some of the needles will penetrate the cells. And when they penetrate the cell, they break, they porate the membrane, and then you have a current flow from the inside of the cell to the outside of the cell. And in this way also, you can record, let's say, action potential that resembles to the one that you get with patch clamp. Okay, so always the reference is patch clamp, but you would like to develop technologies that go beyond this to record the electrical activity of the cells. This is something that is relatively similar to the previous one. Is again an electrode. And this time, rather than putting a needle, you put a sort of what is called a mushroom. And this mushroom, what it makes is that the cell membrane engulfs the mushroom, and eventually it porates, it forms pores, and this is where you can access the inside of the cell. So you need to porate the membrane. So all these techniques record intracellular, let's say, action potentials, but they need to penetrate the cell. They need to break the cell membrane, which is having advantages and disadvantages. But and finally, there is one technique which I would like to mention, which is the nanoscale version of patch clamp. This is a technique which is very close to the techniques that we work. It's called scanning ion conductance microscopy. Scanning ion conductance microscopy is a scanning probe technique. So it's like our techniques, it's a scanning the sample. As a tip, they use a micropipette that has a hole there. And what you do is you measure the current. Oops, okay, doesn't work now. Okay. You measure the current that is flowing through the pipette. Okay, so this is your scanning pipette. Ions will flow. and then it will go out through that. Then, since you are constraining the ions to move in a very narrow area, and when you put the surface of the cell there, the ions, they have a lot of difficulty in getting out. Normally, you don't need to touch the cell, okay? So this increases the resistance. So then by measuring the resistance, you can make topographic images of the cells like we do with the AFM. So scanning ion conductance microscopy is an alternative scanning probe technique that can be used to take topographies of living cells, for instance. It has some issues, but it is a technique that is there. But in addition to this, you can use it like a patch clamp electrode because it's a micropipette. And therefore, what you can do is you can try to attach the cell with this micropipette and then to measure the activity of the ion channels that are in that area. And this is what they have done here. You see, this is single ion in patch clamp, and this is a single ion channel in a scanned pipette. Of course, it is not identical, but it's a real breakthrough to be able to measure with this scanning pipette the activity of an ion channel. Okay? So this technique has been there for many years, but still is not widely used. Okay, so there is a lot of potential because you can address active properties of cells. You can address them at the nanoscale if you want, but it has many drawbacks. And for the moment, it is still there. So maybe there are groups which are more experts on that, but still facing some difficulties. Okay, so this is this is the demonstration that you can record single ion channels by using this scanning micropipette, by using scanning microscopy as a technique here. Okay, so then now just the last 15 minutes, I will be talking on a technique that we have been working together with collaborators in Italy, which is, again, trying to measure active properties. So we would like to measure the electrical activity generated by a cell, for instance, a cardiac cell or a neuron. We would like to use a planar system, not any puncturing system, not destroying the cell. We would like the cell to be there for many days. And we have been working in organic transistors. This is one type of organic transistors. Organic transistors have been there for many years now, but this is a specific type of organic transistor that can work inside an electrolyte. They are called electrolyte gated organic transistors or electrochemical chemical transistor, depending on the material that you use. And the idea basically is you have an electrode here, another electrode there. This is the source, this is the drain of the transistor. So the current that you measure flows between source and drain. And then here you have the organic material, which is semiconductor, is able to conduct electricity. And then at the interface between the organic material and the liquid, there is this accumulation of ions that I mentioned before, that is the electrical double layer. So the electrical double layer is like a capacitor at the end of the day. So basically this double layer insulates the organic transistor from the electrolyte. So then you have here, you have a sort of configuration which is the same as for any transistor. You have a metallic, which is the electrolyte. The interface is the insulator, the organic semiconductor, and the source and the drain. So we know from the physics that if the potential of this gate, which is the electrolyte, changes, the current that flows here changes because the organic semiconductor is sensitive to the potential that you apply to the gate. This is the principle of any field effect transistor. No, you have conduction, then you apply an extra voltage, and you modulate the current between source and drain. Okay, so then here, basically, this is an ideal system to use with living cells. And basically, you have the transistor. Well, these are the electrodes. They have interdigitated. And basically, here you have the cell. These are cardiomyocytes, which has been grown directly on there, just as we do with glass slide. So rather than using a glass slide, we use this one. And then we wanted just to test if the following is true. Here, when there is no cell, I know if I put an electrode here and I change the potential of this electrode, like I did here, I see that the current that flows through there also changes following this. If I put a smaller signal, I get a smaller response. So in principle, reading the current, in some sense, is the same as reading the potential of this space there. So now, what happens if I put a cell, a living cell there? Well, this is a cardiomyocyte. So this cell, they beat because they contract, and every time they contract, they generate an action potential. So they change the potential of the surrounding. And basically, when you don't have cells, you don't see anything, you have just noise. This is the current that flows here. When you put the cells in basal medium, which is the physiological medium for this cardiomyocyte, you start seeing the beating of the cell into the current flowing through the transistor. So it means that it is possible to record the electrical activity of the cell by using this transistor. This is not new in itself because it was already demonstrated. Also, you can do this with a microelectrode. You don't need a transistor. So what is but before telling you what is different, just to highlight the advantages of using this type of setups, you can do measurements for days. In fact, we did this experiment during 1 month, and the cells were still alive, they were still beating, you were still recording signals, etc. This is something that with patch clamp you cannot do, and with microelectrodes you can do. Okay. You also can do tests of drugs if you want to test the effect of a drug into the cell. So you can put a norepinephrine into the solution. This interacts with the cell, and in this case, it increases the beating rate of the cell, and then you record an increasing beating rate. Okay, so you can do pharmacology if you want with this type of setups also. So what is the novelty of all these of these things? Well, the novelty is that in a later work done basically by Adrika Kindiach in the Italian Institute of Technology, they have investigated further the shape of the signal that they were recording, and they discovered that the shape of that signals is very much like the one that you get with patch clamp. And this was a really surprise that we didn't identify the first time we did this recording in our lab. And the idea that we understood is that if this is the cell sitting on top of the transistor, if you cut it by half, you see this is a cross section, this is the internal part, and this is the contact between the cell and the organic transistor. It's practically a direct contact. And then if you zoom still there, what we realize is that the contact here was extremely good. So the cell membrane apparently didn't broke down, it's still intact, but it is very tightly attached to the surface. And therefore, we demonstrated that theoretically, that if these conditions are met, the signal that the transistor will record will be pretty much similar to the one that you record with a patch clamp, but breaking the membrane. This is a very strange situation that has been predicted to occur also in other systems, but indeed, by using an organic transistor is really reproducible. You can use it very time. And here it is, the demonstration. This is the signal that we record with the transistor. You see, it has a shape. Here there is one example, just to show the shape. And this is the signal that you record with patch clamp, which is the reference for this system. And then you see, it is not identical, of course, but it's pretty much similar to the one that you have with patch clamp, okay? And this can be done, experiment in patch clamp you can do for a few hours. In our case, we can do this for months. So that's a big advantage from the point of view of the application. If we compare this, what you get with just electrodes, MEAS is micro electrode array. This is a standard recording systems of the electrical activity of cells. It's just planar microelectrodes sitting on a substrate and the cells on top of it. The signals that you get are these, and you see that the shape of this has nothing to do with the shape of that and nothing to do with the shape of that. The reason is because this is what is called a field potential, is the extracellular potential only. This is an intracellular potential. And apparently the transistor is recording something which is closer to an intracellular potential, even if we are not penetrating the cell membrane, apparently. Okay. So this is the main result that it was obtained by using this type of technologies. And then, of course, you can do also some pharmacological studies. You can say, okay, what happens if I add again the same drug like before? I expect that the duration of the action potential will shorten. This is the effect that produces this drug. And indeed, this is what is observed here. You see that over the time, this is shortening. It is interesting to know that the response of the cell is not instantaneous. So when you put the drug, it takes some time to feel the drug, react to it, and this can be sometimes even hours. So then doing this experiment in patch clamp is complicated. And so you hardly ever see the transition. So basically see the beginning, and then you do another experiment and you see the end. But since this is working all the time and you don't perturb the cell, you can do it in continuously. And there are other aspects that I will not, but even the fine details of some parts of the action potentials can be recorded with this, which is something which is difficult to record with other techniques. Okay, so this is basically the summary of what I wanted to explain. I didn't want it to be too long because it's a hard morning. So basically the summary is, of course, beyond AFM techniques that we have explained it in the beginning, there are many other techniques that you can use to probe the electrical properties of cells or molecules or whatever. The traditional techniques, so the ones which are well established and most of the people is using, is patch clamp. This is to record active properties, and then microelectrodes arrays, which is used to measure passive properties. So this is the standard. This is what is there. If you would like to use them, there is protocols and standards very well defined. Instead, as alternative to this, there are other techniques which over the years, they are been under development. And in particular, I have shown two examples. There are plenty. Okay, there should be thousands even. So that's I showed two examples to show what is different from using this as compared to new technologies. Okay, for instance, on the one side, I showed on-chip technologies. This is something that goes in the direction of what is called laboratories on a chip. So the idea is that rather than using big instruments to do different experiments, trying to integrate everything into a chip and doing the same experiments, but in a controlled environment, very cheap, very fast, etc. And one example I showed is this impedance flow cytometer, which can be used to count cells, to size the cells, and even to distinguish some different types of cells. And finally, I have shown different types of systems as alternative to patch clamp, and I focus more on that system, which is the one that we have been working in the last time, because I think it has a lot of potential, because in principle it's able to provide a fidelity to the signal similar to patch clamp without the drawback of affecting the cell function. Okay. Time has to tell if this is true or if we are going to find some drawbacks in the next year and we will have to not to use anymore. But for the moment, everything seems to be aligned, and we can do these long time experiments similar to patch clamp. And these instruments are already tested by groups working in medical doctors working in hospitals, and they say apparently it works. Who knows if this will work forever or just that after some time it will not work anymore. Okay, so that was it. Yeah. Questions? Yeah. At principle, they have similar problems. The only advantage, one of the advantages is a planar instrument is much easier to handle as compared to an instrument handled by micropositioners. So even if it has the same issues, the experiment is much easier. So you just need to plate the cells on top of this substrate and you forget. You don't need even an optical microscope. You don't need micropositioners. You don't need someone which is expert in punching the cell. So from the point of view of using it, it will be already an advantage. Okay. Then the major advantages, and this is why they would like to do this, is that at the end, you can address the different electrodes independently. If you do a nice array of electrodes and a nice design. So then rather than measuring just in one point, you could be able to map, for instance, all the points in the cell, or, and this is why this has been done, if you have a network of neurons, for instance, then you can just put a substrate, plenty of these, and then you can track how the electrical signals are moving from one neuron to the other, what is the shape, if the shape changes or not. All of this is something that you cannot do with PatchCam. So there is plenty, and this is the main motivation of this group is everything which is planar means that you can scale up later. And if you do one transistor, then you can put 1,000. So then you increase the spatial, you can map spatially, you can do time propagation experiments, which are difficult to do with PatchCam, etc. So even if they have this limitation, still they are very useful for electrophysiology and electrical studies.
Thank you. Okay, so let's start this presentation. So before, yesterday there was a question about forces, long range and short range. Also, this morning appears the concept in the presentation of Gabriel. So actually, I wanted to say something because maybe a bit confusing. When we are talking about interatomic forces between one atom and another atom, a molecule and another molecule, the definition of long range and short range is like this. Below 5 angstroms is short range. Above 5 angstroms is long range. So van der Waals forces at 1 nanometer or 2 nanometers are called long range. That's one thing. Now, if you are talking about electrostatic interactions, magnetic interactions, the definition of long range is different. It could be 100 nanometers, 1 micrometer. Because there you don't talk about atomic interactions. You are talking about nanoscale or macrometer objects. Okay, you are talking about interaction between macrometer scale objects. Then long range has a different meaning, longer, that when we are talking about interatomic interaction, where people in the book, in the textbook, you see long range 1 nanometer or 2 nanometer. Okay. So now, yesterday, what I presented was a tutorial of some established knowledge about AFM and about nanomechanical mapping by AFM. So what I will give today is more research topics that I and my group are working on. And actually the research topics are three-dimensional AFM, high-speed bimodal AFM, and cell mechanobiology. And we are apply or develop tools based on AFM to really address problems on solid liquid interfaces and nanomechanical properties in molecular biology and mechanobiology. So the first, an introduction, why solid liquid interfaces are relevant, or water solid interfaces. Well, The first thing is that water is essential for our life, but it's not only because it brings the nutrients to our body. It's also because the interaction of the water molecules with the proteins was touched this morning lightly, help to stabilize the three-dimensional conformation of proteins. But in addition to this property, also the interaction of liquid with solid interfaces is key to develop some technologies that help us to make the world more sustainable, like process for carbon dioxide reduction, or to generate user-friendly batteries, and batteries long-lasting, and they don't develop waste or toxic chemicals. So for this, we developed several years ago a concept which is called three-dimensional AFM. And in three-dimensional AFM, what you are having is the tip that is oscillating, and at the same time that oscillates is moving in the three spatial coordinates. So the idea here is to develop a system which is so sensitive that the passing of a single solvent molecule modifies some of the dynamic properties of the oscillating of the tip that I presented yesterday, the amplitude, the phase shift, and we could generate a three-dimensional map, a volume map of the solid-liquid interfaces with atomic scale resolution. So that was the concept, and this is the realization. Actually, some of you yesterday saw a picture of the microscope. So actually, this is the fluid cell, and actually the tip, this is the tip, the cantilever with the tip. So you can see that our volume is very small, 20 microliters, but macro scale. We can see it with our eyes. So that's how they look like, and actually we have several generations of this dimensional AFM, and the latest generation can perform measurements in situ and under voltage or electrochemical conditions. And this is how it looks like, the system. So what we do is we generate sequentially planes in the YC direction or in the XY direction, and then we move the tip to another Y position to generate a different plane, and then we combine this sequence of planes to generate a volume image of the interface. And what is key for this system to work is, well, these are the observables of the system, the amplitude of the oscillation, and the phase shift between the force, the driving force, which is photothermal, and the tip response. And well, those observables can be transformed by some theory in forces, and those forces reflect the different organization of the solvent molecules near the solid surface. But what is key? What is key to get molecular resolution in a liquid? Let's say the overall diameter of a water molecule is 3 angstroms. So in order to see a water molecule, you need that the oscillation of the tip is 1 order or 2 orders of magnitude smaller than the size of the molecule you want to see. So the key is that these oscillations that you see here are in the 40 picometer regime. But it's not only that the total amplitude is 40 picometers, it's that you have to be able to see changes of 2 picometers. That's very small. That's very small. Okay. So this is the key for the system to work. Of course, there is some physics and electronics behind that I will skip. And once you have accomplished that, this is a interface of mica, a very friendly substrate for biological studies with AFM, with a solution 200 millimolar of potassium chloride. And you can see here the mica is in orange, and you can see in blue, say, the bulk water, and then you see these layers of water molecule. The thickness of this layer is just 3 angstroms. It's just the ordering of some water molecules. So the water molecules near a solid surface are ordered. is no longer the same disorder that we have in the bulk. Always in the near the wall of a solid surface, glass, when it's surface, the liquid molecules tend to be organized. And then you make a cross section, you can see here the absorption of the ions of the potassium on the surface and how the water molecules are structured. This is an animation, so it shows you how the things work. So actually, well, we have been working with this system already for 8 years now. And the first really real funding that we discovered with this instrument is just the organization of water on hydrophobic surfaces. So actually, what we get are these type of frames, the bulk liquid, and the layering of the liquid solvent molecules near the surface. And actually, what we are going to, this is the force as a function of the distance. You see very small distance, 2 nanometers. And then we get, you get the oscillations of the force depending on the organization until we get into contact with the mica surface. And what we are going to plot in what is follow is just how this distance between these two peaks, this layer and this other layer, changes with the surface that we use. And we are going to study the origin of hydrophobicity. What is really, what means hydrophobicity at the molecular scale? So at the macro scale, we know what a hydrophobic surface is. So when we put a drop on a hydrophobic surface, we get a drop. However, if we just put this 20 microliter drop on a hydrophilic surface, the drop spreads out, tends to wet the surface. That's the macroscopic visualization of hydrophobicity, hydrophilicity. So now we did these studies on a mica surface, which is a prototype hydrophilic surface. So I think this is a video. And we see the atoms of the mica, the atomic lattice, and the structuring of the ions that are absorbed, and the layering of water that also has molecular resolution. I didn't show here. Well, the thing is that when we measure the distance between the layers, we get 3 angstrom, which is what it should happen for the size of the water molecule, and it fits molecular dynamic simulations and everything. So the idea is that when you put water in contact with a hydrophilic surface, the layering of the water is 3 angstrom, which is what it should be. So no, and this has been applied also to biological molecules like collagen nanofibers, and we get again this layering of the water on top of the collagen nanofibers. Another type of substrate, so this is has some steps, and the layering follows the steps nicely. Well, we have really demonstrated this, that any hydrophilic surface, even the one that had been functionalized, they show this layering of 3 angstroms. So now, let's do an experiment on some hydrophobic surfaces, or mildly hydrophobic surfaces like graphite, graphene, or any type of 2D materials like molybdenum disulfide, tungsten diselenide, hexagonal boron nitride. So what do we get? Again, you can see that this 3-dimensional AFM preserves the spatial resolution, the atomic resolution of the AFM, plus the layering of the liquid. And we get the same, practically similar images in appearance with the one that we obtained on mica. Sorry. And this is for the other samples. Anything. So what is really relevant is that when we measure the distances between the layers, we didn't find the 3 angstroms. We got 4.3 to 5 angstroms. And this, at this level of resolution and accuracy, is a huge thing. So going from 3 angstrom to 5 angstrom is like going to a different scale. So the thing is that water cannot be layered and be separated by 5 angstroms, because this is does not scale with the size of the molecule. So the thing is that water is not forming these layers that appear when we introduce ultra pure water, or we deposit ultra pure water. This was ultra pure water, purified water, on these materials, these hydrophobic materials. So what has happened? So and also because we have the capabilities of imaging, we can get an image, of course, on the graphite, and we see the graphite lattice, but also we can get a two-dimensional image here on the layer, or here. That's something that the instrument, when you use it very nicely, can do. And actually, when you do that, you get these parallel structures. And when you go to the molecular resolution, these structures are separated by 5 angstrom, which was actually the size of the layer. But this, so what we are looking here is not in the perpendicular, which is this direction, but this direction, just inside the layer. How the molecules which are there, that they were supposed to be water, but they are not water, are separated, and they are 5 angstrom. So what are those molecules? Actually, those molecules, well, we have done these studies with molecular dynamic simulations, some specific Raman spectroscopy. So actually, the layering is formed by hydrocarbons. Hydrocarbons, which are in this room here, there are traces of hydrocarbons, very small. You count 1 billion. You count 10 billions of nitrogen molecules in the room, 1, 2, 10 billions. Every 10 billions, you count 1 hydrocarbon. So that's nothing. However, what happens is that thermodynamically, even that the amount of these hydrocarbons in the water or in the environment is really negligible, trace amounts, what happens is that these hydrocarbons love to be sit near a hydrophobic surface. So when they are in the water, they diffuse. Eventually, they reach the hydrophobic surface and they stick to it because the free energy is favorable. You see, they have a minimum. And then in the process, they are replacing, expelling the water molecules. So at the end of the day, if you have a pencil, which is made of graphite, and you dip into your water bottle or in a glass of water, the water molecules will never be in touch on average with the graphite. There will be a barrier of hydrocarbons of 1 nanometer that will be between separate the water and the graphite. So that's a discovery that we made with this instrument. So this is the scheme. So at the beginning, you have the water in contact with the hydrophobic surface, but eventually these hydrocarbons diffuse. And when they go to the surface, they stick and then expel the water molecule from the interface. Of course, if you do this experiment with a hydrophilic surface in the water, there is also these hydrocarbons. But what happened? That when the hydrocarbon diffuse and go to the hydrophilic surface, the mica doesn't like the hydrocarbon. So the hydrocarbon goes there and moves away. And the water molecules stay there because the mica likes more the interaction. This is expressed, this likeness is expressed in terms of free energies. So that's the conclusion, that the same water structures difference on a hydrophilic surface, on a hydrophobic surface. So, well, that's the first topic. So the other topic that we are working on is high-speed AFM. And actually, this is an editorial from Nature Nanotechnology, 2016, what was the 30th anniversary of the invention of the AFM. And then they pointed out that in the AFM, after 30 years, there were still some active fields of research, and they mentioned high-speed AFM and multi-frequency AFM. So actually, high-speed AFM has been an advancement of the AFM that was, I mean, now is when is the realization of the high-speed AFM, but from the beginning, the pioneers of the AFM, Paul Hasma group, for example, that was a leading scientist driving the use of the AFM in biology, they realized the limitation of the AFM, which is actually part of the motivation of this network. The low throughput, the slowness of the imaging. I mean, that was realized 30 years ago. It's not new. The problem is that the solutions were not there. So the goal, in particular for surface science, molecular biology, is really to have a technique with this atomic or molecular scale resolution, but also that images processes when the processes are happening in real time. That way, high-speed AFM is needed. And actually, the scientists and the group that was able to solve all the technical issues regarding the use of the AFM for high speed was Toshio Ando groups in Kanazawa University. And actually, this is an iconic image of what high speed is about. So this is the working of a myosin 5, a protein that assembles an actin filament. So it's working an actin filament which are in the cytoskeleton of cells. So you can see how it's working. And these are 10, I mean, the resolution, the frame is 30 nanometers. So this is almost 1 or 2 nanometers spatial resolution. And the frames are milliseconds. So that was the first time that really we were able to capture, even there was some ideal conditions, to capture really the working of the interaction of a protein with another protein, another fiber in real time as it was working, and they were able to express or to show how this interaction was happening with this molecular detail. So that was the, and actually, but so what is the key to high, because particularly I am a more instrumentation scientist, what is the key for that? Of course, Ando's group realized that there were many items, many issues that need to be improved to make high-speed AFM. The beam detector, the amplitude detector, high-speed AFM is performing amplitude modulation or tapping mode AFM. The proportional integrate the controller, how driving the piezo, the scanner, etc. Actually, in each issue, it was a 1, 2, or 3 PhD thesis. Okay, so it took a lot of time and a lot of PhD thesis to develop high-speed AFM for them and also for other groups. And the key issues are we have to use a small cantilever, fast amplitude and phase detection, fast C actuator, so on. So actually, I am going to illustrate one of the issues of the cantilever size and the speed, and why high-speed AFM needs to be performed with small cantilevers. So this is a standard cantilever for tapping mode, 100 micrometers in length and 30 micrometers in thickness, and this is a cantilever for high speed, 10 micrometers in length and hundreds of micrometers in thickness. Okay, so what is the key? If we calculate the resonance frequency depends on the material, the Young modulus, the density, but on the ratio between the thickness and the length. So high-speed AFM requires very short cantilevers because high-speed AFM needs high frequencies. Not that high-speed AFM asks for short cantilevers. High-speed AFM asks for very high resonances. and the high resonances are given by small cantilevers because this geometry. But why we need high frequency resonances for the cantilever to do high speed? So that's the question. Why? Well, there are several reasons. The one reason is that the cantilever introduces a mechanical bandwidth, which is this quantity here. And this mechanical bandwidth is proportional to the resonance frequency. So the higher the resonance frequency, your mechanical bandwidth is higher. So the system can respond to higher frequencies. When you have a slower resonance, the mechanical bandwidth decreases, and then you are slower. Okay? Well, the physics behind this is complicated. It was something that yesterday I put super advanced. So I will skip it, but if somebody is interested, I can answer. Okay, but it's behind the super advanced. And the other thing here, but well, this illustrates a little bit. So at the end, what happens intuitively? You know, this is the resonance, the first resonance. So the high speed AFM, you have to move everything fast. But this thing fast has to be, for the cantilever, has to be at this frequency here, far from the resonance. Why is that? Because if you excite something, the cantilever of a force, with a frequency close to the resonance, the cantilever is going to amplify that thing. And that amplification is noise. So the cantilever doesn't need to know that you are really moving it fast. So that's why you have to move the tip here at this frequency far from your resonance. So the cantilever doesn't see, the resonance doesn't see that you are acting on it. It's dampening it. But if you were exciting or moving the tip at this frequency, the cantilever will see and will amplify, and that will create a lot of mess. So we combine this with the bimodal AFM that I explained yesterday. That is a method to get mechanical properties or other kind of properties, electrical, magnetic, without getting a force distance curve, but just measuring or sensing how the interaction is modifying the amplitudes and the phase shift. Oh, sorry. Okay, so this is the concept in bimodal AFM, is that we have the cantilever and we excite it with two frequencies. The first resonance, the fundamental resonance, the resonance that we usually talk about when we talk about cantilevers, and the second resonance. And then you get a complex excitation, and then you get a complex response, and then what you do is you separate with locking amplifiers the components of the first resonance with amplitude and phase shift, and the second. And then there is a theory that connects the changes of the amplitudes and phase shift with mechanical properties, following the schemes that I presented yesterday. And once you have that, well, then you can generate maps of mechanical properties with of proteins in liquid, so in buffer solution, with very high spatial resolution, and just mapping the changes of in this case, the jam modulus of this proteasome, and then you can link the why this proteasome has to be stiffer here and here than in the middle, and you can correlate these properties with the functionality of this material. And then bimodal AFM can give you molecular resolution in other systems, no problem. So the idea is to combine bimodal AFM with high speed, and that's something that it can be done. And for that, well, we have to implement in the bimodal AFM the concepts of some of the technology developed by Toshio Ando to get amplitude detector with a smaller noise, with a higher bandwidth, and the use of smaller cantilevers, and all the technology of high speed, actually. So actually, this is a comparison of really standard AFM imaging, 1 Hertz per line, fast AFM imaging, 10 Hertz per line, and a high-speed AFM, which is, this is like 200, well, in this particular case, it was 200 Hz for line. So the sample is the same. It's a PMMA PS diblock copolymer. So you can see really that the high speed means something. In this particular case, to have high speed bimodal, because actually these are maps of mechanical properties, not topography. Yes, but in this particular case, that the sample is passive, still high speed AFM only enables us to get images faster, but the same quality could be obtained if we did the experiments with a regular film. So the point of high speed AFM is to go into dynamics. So again, this is an experiment that you can really get different properties of the system at 5 frames per second, which is more or less close to the limit of what we can do with our instrument right now. So now we are going to study the dynamics of the self-assembly in the early stages of collagen molecule. Collagen molecule is, well, is the most abundant protein in our body. It's usually in the extracellular matrix. It forms part of the tissues, the tendons, the bones. Well, it's present in many parts of our body, and also is a component of hydrogels. I think some of you or the other day were presenting some work involving hydrogels made of collagen. So what it forms a structure, a nanofiber structure, and we apply this bimodal AFM to see how the process of the structure of the molecule of the nanofibers happens as a function of time, and you can measure simultaneously changes in the topography, the height, in the elastic modulus, and in the viscosity properties of the loss tangent. And you can see how the molecules look for it, interact. So this is the kind of thing that high speed in combination with this nanomechanical mapping of bimodal AFM can give to molecular biology or polymer sciences. Well, this is another image, is more interesting because we see this polymer, the unit length is 300 nanometers. So actually you see the tip, and the tip increases by 300 because it attaches this collagen unit, which is the topocollagen is 300 nanometers. You just see, you see the tip, and eventually at some point grows 300, and then 300, and then is forming the structure that is the D band. And why is this thing moving this way? This is the thermal noise. The thermal noise of the molecule on the surface is what is making this fluctuation, and actually is the thermal noise which is driving at the end the formation of the fibers, because that's what they get the energy to move around the surface and to find the other collagen and just to do the structuring. Well, so the last topic is just in the context of mechanobiology. So the context of mechanobiology has been already illustrated. Well, is key to the project, to why we want to develop tools, AFM tools for life sciences, is because it has been discovered that really mechanical properties are key for biological function at the cell level. Well, we knew that from the beginning because we are, I mean, if I drop here, I could break my whatever, and then it's a mechanical force that damage my body. Okay, but what is mechanical biology, not this macroscopic observation, is that at the single cell level, the mechanical stresses drives the behavior of the cell in a subtle manner, and this was demonstrated years ago, in 2006, by a breakthrough paper of Dennis Disher, where they got stem cells, so cells that have not differentiated, and they put stem cells in different substrates. A stiff surface, like a petri dish or glass, and then a softer surface, like a hydrogel. And then they realized that the differentiation, the same stem cell culture on a stiff surface mature into a stiff tissue, like bone. But that cell, that stem cell, it was cultured in a soft substrate, mature into a neuron, a very soft cell. So the interaction that the receptors of the cell have with the surface for the attachment drove were translated to the nucleus and modified the expression of the genes of the stem cells to differentiate in one direction or the other. This is mechanobiology. And the AFM, being a mechanical microscope, is ideally suited to address issues in mechanobiology. And the cell biologist knows that. And in particularly, we are collaborating with a lot of groups on this topic. So this is how the AFBAT, as the project coordinated by Gabriel Gomila knows it, there are many things that need to be solved to make the AFM, and that's why you are here. You are supposed to really to lead the way in the future to make the AFM to go from a laboratory instrument to a instrument for a hospital, for example, like NMR. And well, this is how the AFM operates. Have the tip, and we are just interacting and getting 4 distance curves. And the 4 distance curves depend on the region of the tip. Are this way, and in this way you can get nanomechanical maps, which was what we put the basis yesterday. And of course, yeah, there are, so then the idea is to get these 4 distance curves, and the 4 distance curve have a signature of the local region that you are exploring. So the cells are really tricky in many ways because, well, first is one of the softest physical systems on Earth, solid system, but it's a complex system that has solid materials, lipid bilayers, DNA, proteins, but also a lot of water. In a cell, in a mammalian cell, 70% of the weight is water. That means viscoelastic properties as well. A cell evolves, it's a living system. So when you are doing the experiments, the ones of you that are working there, you know that after 1 hour, probably your culture, the cells could be dead. Has a finite thickness, which is a problem for contact mechanics model, and it's a viscoelastic. So actually, why is viscoelastic? Well, we know that. You take a curve approaching, you go this red line. If you withdraw the tip, you go the blue line, and then the area in between is the energy that was dissipated due to viscoelasticity. We developed this general viscoelasticity theory that I will not explain now, but we develop it for any asymmetric tip, which this is the theme. Of course, these are mathematical expressions. The idea is really to get into analytical expressions that are easy to handle. We managed to do that with several hypotheses that I will not describe that, but they include the finite thickness system, and they include something called the things model, because when we approach the tip, we go one way, and we go the other. We go back. We go in a different pathway, and then teams in the 60s develop a model to relate the state of the system when you are approaching and you are retracting the contact area, and that was implemented for this theory. So the question really, what is here? Okay, now, this general theory requires this quantity, which is called relaxation function, which is the quantity that describes the viscoelastic response of your system, in particular the cell. So the point is this, okay, there are many viscoelastic models out there. Which is the one that we should use with cells? Well, the first thing is that you have to distinguish mammalian cells from bacteria. They are different. They are called cells, but they don't belong to the cell group. So we are working with mammalian cells, which are the ones that are in our bodies. So mammalian cells are very soft. Bacterial cells, I mean, somebody here was working on bacteria. Bacteria, the young modulus is of the order of gigapascal. So it's a different material mechanical with respect to a mammalian cell, which is 1 kilopascal. So many orders of magnitude in between. So which model works? Well, the model that works is what is called 3-power law rheology. And this model has two components. One is the realization function of power law rheology, and the other, the geometry of the tip. And when you combine a realization function with a geometry of the interaction, you get the viscoelastic model for the AFM. That is called three-dimensional because you include the geometry. And the geometry are in these coefficients, beta and alpha. Those are the coefficients that bring geometry into the expression of the force. Okay, so power law rheology. Well, there are different ways of deducing this power law. Well, the first thing, what is power law rheology model? Well, it's a model that has a realization function in this way, which depends on two parameters. The modulus, which asymptotically coincides with the Young modulus, and an exponent, which is called fluidity coefficient. And the fluidity coefficient changes between 0, elastic solid, and 1, a Newtonian viscous liquid, an ideal liquid. And for a cell, you will be in between. Well, this model theoretically can be deduced in different ways. Some groups have deduced it by using putting the constitutive equation in terms of fractional derivatives. You are very mathematical oriented. You like to do that. But other people, more physically oriented, thought about this model like you have a system with the components in different potential wells, and they are randomly distributed. And when you do the average, you get a power law response. But you can also deduce this model by just considering a spring and a dashpot, a series of infinite springs, spring and dashpot, and do an asymptotic expansion, and you can get also a power law. So different ways to get a different result, to the same result. Why it works? Okay, so this is the thing. In black is the experiment performed on HeLa cell. Well, we have done it in many mammalian cells. And in red is the fitting. Look, I mean, I assure you, the same model fits two different for distance curves. The model has two parameters. You cannot do better. To fit two different expressions of the force with two parameters, you cannot do anything better. That's the most optimal thing. Because you know that you can fit a curve with anything. You put a polymer with many coefficients and you fit everything. So that's the question is not that you fit. The question is that you fit with something that is sensible and also that has the minimum thing. So it works. And then, of course, you can represent the values of the modulus as a function of the biological condition and the fluidity. So one could think, okay, what would happen if I use a different viscoelastic model to fit the experimental data? You can do it. This is the Kelvin Boy case. So you got, I mean, here we cut the curve here because some limitation here, but it's the same thing. You are fitting these things. So this is the experiment. Now you defeating with the Kelvin Boy. You don't get it. You could fit maybe one part of the curve, but not the other. So that's the key point. So now, and this is just the last part, and it's also the part that related heavily to the machine learning side of the SPM 4.0, is that we want to improve the speed of imaging, making better instruments, better AFMs, faster. But also we want to improve the throughput. So the time that takes us to get data from the images. And that's where machine learning can work. Because, for example, if I generate this nanomechanical map of this fibroblast, it took the people that work with me 8 hours to put this map in terms of mechanical properties by using the standard process, which is nonlinear least square fittings. I think yesterday Jaime, to some of you, showed something about that. So 8 hours, I mean, that's a lot. That's a lot of time, but that's what usually graduate students, I was a graduate student at some point, it took us to do experiments to analyze data. So that's not scandalous for a graduate student. But if we are planning to get the instrument in a hospital, cannot be. This is impossible, unfeasible. So that's where then we develop a process to really process for distance curve by using machine learning. As I said, least square fitting works very well, very slow. And machine learning works very well, very fast. And this is something, so I will not describe what we did. Of course, we use synthetic curves because for the training, because using experimental curves for the training would be very time consuming and also unreliable because cells are different. And we combine these neural networks made of two neural networks, actually, one for the modulus and the other for the fluidity coefficient, because it's based on the power law rheology model. And the bottom line is that really to get this nanomechanical map by using this neural network took us 40 minutes. And by using the standard with the same computer, we didn't change the computer or the CPU or GPU, the same actually computer of the instrument, which is a GPK instrument. We went from almost 9 hours to 40 minutes. So that's a major step that really we think is very good in the process of reducing the throughput. So yeah, as summary is that in the topics of advanced AFM method we are working is just to really develop new tools to study solid liquid interfaces with atomic and molecular resolution, and we have really pushed the AFM to a level where no other technique, spectroscopic technique, can do. So then we are just combining AFM with the multi-frequency approaches, the bimodal AFM, to get a nanomechanical mapping of proteins in real time. And now we are also working in cell mechanobiology just to learn more the physics of cells, and by learning the physics of cells to really to develop tools to apply to mechanobiology and medicine using machine learning approaches. So these are some members of the group evolves, changes, and some people are here, others move out, or come in. So I have to put different pictures because I cannot have everything the same time in the lab for some reasons. And also we have some doctoral candidates from other positions. Well, and thank you for your attention.